Hi, my name is Wendy Demas. I represent the Rat Genome Database, RGD, at the Medical College of Wisconsin. RGD joined the Genome Reference Consortium, GRC, to further improvements of the mammalian rat, brown Norway, version 7.2 genome assembly through the curation of errors brought forward by RefC curators in the rat community. Today, I'm going to describe the workflow used to resolve those reported issues. The genome records were annotated with the NCBI eukaryotic genome annotation pipeline. Assembly issues are recorded in the Atlassian JIRA ticketing system. The GRC RAT issues dashboard pictured here provides public access to individual views of all reported issues and their resolution status. To determine a starting point, RGD provided the summary of issues to members of the International Rat Omics Consortium with a request to identify specific genes of interest in the community. These genes were used as a filter to prioritize tickets with reported known or potential effects on protein function. The resolution workflow is broken into pre-assessment and resolution stages. A variety of tools provided by NCBI, RGD, and the Genome Reference Consortium are utilized to review and resolve those tickets. RG129 is reported to have a 1 nucleotide insertion, which results in the frame shift in the encoded CDS and premature termination of the protein. The first step in the pre-assessment stage is to identify the strain listed as the source in the NCBI nucleotide database. In this ticket, a specific strain is not provided. The reported position is reviewed in RGD's variant visualizer tool. These data may indicate if the reported issue is common across strains, as seen here in RG129. The inset box indicates that all reads for this position do not have an insertion for all strains. Illumina short read data of the same animal used for reference generation are viewed in JBrowse. These data serve as a comparison to the long read data used to generate the reference. This step allows the curator to compare different reference assemblies and potentially identify problematic assembly regions. The global view shows some assembly issues from reference version 6 may have been corrected in reference 7.2. The long read alignments are viewed in the NCBI Genome Workbench tool. This tool is where the heavy lifting of the resolution occurs. Alignment, reference, versions 6 and 7 are easily compared. In this example, you can see the reported issue occurs in a repetitive region. If an alternative alignment was available, it would be visible below the transcript sequence. However, there are no alternatives to review for this region. Since the transcript sequence is stable, contigs for assembly version 6 and 7.2 were blasted against the transcript. This view shows there is a discrepancy in the number of Cs in the region between bills, as well as a mismatch and an insertion in reference version 6. Due to the repetitive nature of this region, this is likely an error in the reference. The issue could potentially be resolved with sequence from reference version 6, but is currently stalled. The transcript for OPRM1 was reported to have an insertion in the new genome relative to the old, resulting in a frame shift for ticket RG8. The strain is listed as Sprague-Dawley, which may indicate the reported issue could be specific to the brown Norway strain. The reported position is reviewed in RGD's variant visualizer tool. The brown Norway strain is highlighted here in the red box. Other strains also carry the same nucleotide for this location. This may indicate a technical error, as all strains agree at this location. JBrowse short data on the left show that the reference version 6 has fewer assembly errors as compared to reference version 7.2. The long read alignments as viewed in Genome Workbench on the right shows an alternative contig is available to potentially resolve this issue. Blast alignments to the chromosomal region show 100% identity between the all contig and the chromosome region of the reported issue. The positive slope of the line in the dot matrix view indicates the chromosome and contig are perfectly aligned and in the same orientation. The alternative contig was also blasted against the transcript. And you see a similar dot matrix view with 100% identity. Individual blasts of the reference 7.2 alternative contigs and of the reference version 6 contig also agree with the transcript. The alignment of the two contigs for reference version 7.2 show agreement with the exception of the reported error. This supports a region of the alternative contig can be utilized to electronically resolve the reported issue. The TPF file was successfully submitted. The gray ball on the TPF view shows the region of the alternative contig is listed as a contained subsequence. Ticket RG8 was closed and the status is updated on the GRC webpage. Ticket RG90 is reported to have numerous indels and mismatches compared to transcript NM0129 19.3, which causes frame shifts in the encoded protein. The results of the first step of the pre-assessment stage indicates the reference is based on the Sprague-Dalley strain. Variant Visualizer provides data for 6 of the 7 issues reported in ticket RG90. Data shows there are variants both common across strains and unique to the brown Norway strain. 4 of the reported issues listed in ticket RG90 will be resolved using the workflow described for RG8. 3 issues in ticket RG90 highlight additional data, such as other strain genome assemblies, would be good evidence to address the resolution of these errors. Preliminary data for this issue, labeled as D, show the short read assembly is better for reference version 7.2. The error was reported for transcript NM012919.3, but other transcripts do not contain this reported missing A. And both the primary and alternative alignment data agree with these other transcript sequences. No variant data are available for this position, and this particular issue is stalled until more data are available. Variant Visualizer shows brown Norway may be unique for these 2 issues listed in RG90. JBrow's review indicates that this region is not overly problematic to assemble. Alignment data for the current and previous reference versions do not agree with the multiple rough transcript sequences. Additional strain genome assemblies would be helpful to determine if these errors are indeed unique to the brown Norway strain. Since some of the errors in this ticket cannot be resolved, the status will remain listed as stalled until these 3 points can be addressed. Thank you for your time and attention.